Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. In this video, we will discuss anomalous secondary growth seen in plants. So it is a very special video because I am uploading this video on a special request from one of my subscribers. So here it is. And this part is also present in your SSB Botany Lectureship syllabus. Anomalous secondary growth of stem and root, Bignonia, Borevia mirabilis, Nyctanthus and Dracena. So today I will discuss this part of your syllabus. Let's start. So in many plants, the pattern of secondary growth or secondary thickening shows deviation from the normal type. At first, what is primary growth and secondary growth? Primary growth of the plant, in which plant is developed from the apical stem or by the help of the apical meristem, and the plant grow by its length in primary growth. In case of primary growth, the plant increase in length, but in case of secondary growth, it increase by the diameter or girth. So the primary growth, where the plant increase by length, occur by the help of apical meristem, but the secondary growth, where the plant increase by the girth or diameter, it occur by the lateral meristem or the cambium. So normally plants, generally dicot plants, show the secondary growth because they have cambium. But in some plants, the pattern of secondary growth shows deviation from normal type. It shows some deviations or variation. The anomalous secondary growth is more common in tropical plants as compared to temperate plants. Anomalous secondary thickening is not an anomaly or disease. Rather, it is an adaptation to suit or adapt the habit or any habitat of the plant. The dicot vascular bundles are conjoined, collateral, open and arranged in a ring. So what is the meaning of conjoint? Conjoint means the xylem and phloem lie together in the vascular bundle. And collateral means the xylem and phloem lie in the same radius. So, and the xylem towards the inside and phloem towards outside. The phloem and it is the xylem. So they are arranged in a ring-like manner and they are conjoined, where the xylem and phloem lie together, and collateral is the xylem and phloem present in the same radius, xylem present inwardly or inner to the phloem and phloem present in the outer layer. In the monocot, the vascular bundle are conjoined, collateral but closed. In dicot vascular bundles, between xylem and phloem, one cambium layer is present, which helps in the secondary growth. The secondary growth starts from this cambium. Formation of secondary tissue takes place by fascicular cambium in the stele. This is the fascicular cambium. and core cambium in the cortex. In the cortex region, the secondary growth takes place by the help of core cambium. But in vascular tissues, the fascicular cambium, these are the fascicular cambium which are present between the xylem and phloem bundle. The secondary growth starts from here. At first, the first stage of secondary growth, the joining of this fascicular cambium forming interfascicular cambium. So these are the interfascicular cambium which joins to cambium or fascicular cambium. But in case of monocot, the vascular bundles are conjoined collateral but closed because they lack cambium. So they lack the secondary growth. And they are in monocot, the vascular bundles or the xylem phloem all are scattered throughout the ground tissue and secondary growth is absent. Any deviation in the above pattern of development, so it is the normal case of the growth, secondary growth. Any deviation in the above pattern of development in primary as well as secondary structure called anomalous secondary growth. So what is the reason for anomalous secondary growth in plant? The normal cambium behaves peculiarly and irregularly, resulting in abnormal arrangement of vascular tissue. Generally, cambium produce phloem towards outside and xylem towards inside, and they show a very systematic pattern of growth. But sometimes it grows or divide irregularly, resulting in abnormal arrangement of vascular tissue. Second, the normal cambium is situated in abnormal position. So at first, they show if they show any irregular or abnormal activity. Secondly, if the cambium is situated in abnormal position. Third one, the normal cambium does not develop, or if present, it is replaced by additional or accessory cambial ring. So the normal cambium at its place is not developed but replaced by another accessory cambial ring, or generally the normal cambium is present as it is and along with many additional or accessory cambial rings are also present in the plant. So these are the cases of anomalous secondary growth and reason of anomalous secondary growth. Then the anomalous or abnormal secondary growth is often observed due to abnormal position of cambium or abnormal activity of the cambium or abnormal position as well as abnormal behavior of the cambium. So keep this in mind. Then anomalous secondary growth is of two types. One is adaptive anomalous secondary growth. Second one is non-adaptive anomalous secondary growth. If anomalous secondary growth has relation with habit or physiological function, such anomalous secondary growth is called adaptive anomalous secondary growth, where plants show some adaptations or deviation to adapt a new habit or habitat. Then non-adaptive type of anomalous secondary growth means if anomalous secondary growth has no relation with the habit or physiological function, such anomalous secondary growth is called non-adaptive type of anomalous secondary growth. Then the anomalous secondary growth seen in Bignonia stem. So secondary growth in Bignonia stem is not typical of found that of dicot stem. So it is not a typical stem. It shows some deviations. In this plant, the cambium is in normal position but shows abnormal activity. In some plants like Bignonia and other family members of Bignoniaceae family, the cambium is normal in position and activity for the first time, but immediately cuts off different proportions of xylem and phloem at four alternative points in the form of a cross. The cambium produces secondary xylem and phloem in variable quantities. In some segment, much greater amount of xylem than phloem. In other, more phloem than xylem. So generally, when cambium is present or shows normal activity, it produces equal amount of xylem and phloem. But here in case of bignonia, it produces variable quantities of xylem and phloem. So it is a abnormal condition. As a result, characteristic structure is formed with ridge and furrow. The ridges are wider than grooves. Secondary phloem forms the grooves. Parallel strips of bast fibers are formed. Cambium breaks off into number of strips wider than the remain on the top of the ridge and narrower remain the base of the groove. Number of edges formed, usually 4 in number, systematically arranged corresponding in position to larger primary vascular bundle. It is a kind of adaptive anomaly exists in bignonia because of its habitat of habitat that it is a woody climber. So to climb, it shows this type of anomalous. So it is the structure of bignonia stem showing the anomalous secondary growth. So here at 4 diagonal points arranged in a cross-shaped order. It is a cross-shaped order. So formation of secondary xylem is reduced. At this point, the formation of secondary xylem is reduced, and that of secondary phloem correspondingly increases. So that the secondary phloem with transverse bands of bast fibers gets penetrated within the mass of xylem in the form of phloem pockets. So these are the phloem pockets. Phloem pockets are the characteristic features of bignonia. Presence of phloem pockets are the characteristic features of bignonia. So this woody cylinder appears to have 4 longitudinal grooves which become increasingly deeper with the secondary growth. As a result of this, 4 furrows at 4 equidistant points appear in the xylem, extending almost to the pith. The cambium breaks off into number of strips, widest ones occurring opposite to the 4 projecting ridges of wood and narrow ones at the base of the grooves. Cambium is not found in the radial surface. So finally, these 4 phloem pockets are formed. So this is the structure of anomalous secondary growth which seen in case of bignonia stem. So why bignonia shows this kind of activity? And yes, apart from that stem structure, bignonia also show fissured xylem. Fissured xylem only seen in old stem. So that wedges of phloem are formed and thereafter the xylem strand becomes fissured by dilation and cell division in woody parenchyma and pith. So, apart from phloem pocket and presence of fissured xylem, why bignonia shows this kind of activity? Because bignonia is a climber. When stem grows around the support, its inner side gets compressed and outer side gets stretched. Only hard tissue like secondary xylem remains in the central part of the stem during compression and stretching, and the stem would break or crack. To avoid breaking and cracking of the stem, it requires some soft tissue in the central region along with the xylem. Central hard tissue is traversed by 4 phloem patches at 4 diagonal places. This is due to abnormal behavior of cambium. There is a direct relation between the anomalous secondary growth and climbing habit of the plant. Hence, this anomalous secondary growth of Bignonia stem is described as adaptive anomalous secondary growth. To adapt its habit of climbing. So, Bignonia shows adaptive anomalous secondary growth. Then, anomalous secondary growth in Borevia stem. Transverse section of Borevia is circular in outline, reveals the following tissue from outside to inside. At first, epidermis is present, single layered epidermis is present, which with radially elongated cell. Multicellular epidermal hairs arise in some cells. A thick cuticle is present on the epidermis. Some stomata are also present. Then cortex layer is found. Then pericycle. Then vascular system. The vascular bundles are present in 3 rings. In the innermost ring are presence of 2 large vascular bundles. In the middle ring, the number ranges from 6 to 14, while the outermost ring consists of 15 to 20 number of vascular bundles. Vascular bundles of innermost and middle ring are medullary bundles. Vascular bundles all are conjoined, collateral and endark. Endark means the protoxylems are present towards the inside and metaxylem towards the outside. Two vascular bundles of innermost ring are large, oval, lie opposite to each other, with xylem facing towards the center and phloem towards the phloem outwards. Middle ring consists of 16 to 14 small vascular bundles. Vascular bundles of inner and middle rings may show little secondary growth. So, in case of Borevia stem, they have many rings of vascular bundles, 3 rings, 2 innermost, then middle ring, then outermost. So, the vascular bundles are arranged in 3 rings in case of Borevia stem. Then anomalous secondary growth in Acheranthus stem. So, these are the structure of stem. At first the epidermis, then cortex, uh, then the vascular system. In young stem, the vascular bundles are conjoined, collateral, open and endark. But old stem shows the secondary growth. In old stem, the vascular bundle consists of primary phloem, secondary phloem cambium, secondary xylem, conjunctive tissue, included phloem and primary xylem. The primary phloem is crossed and obliterate. Secondary phloem is present in the form of complete ring and consists of sheath tube, companion cell, phloem parenchyma. A cambial strip is present in between secondary xylem and secondary phloem. Secondary xylem and conjunctive tissue are undistinguishable. Large xylem vessels are very clear in thick-walled conjunctive tissue. In conjunctive tissue or prosenchyma are present groups of included phloem and interxylary phloem. Primary phloem is present near the pith. Then pith is well developed and parenchymatous. Then medullary bundles. 2 medullary bundles are present in the pith facing their xylem to each other. So it is also a abnormal condition. They are conjoined, collateral, open and endark. So presence of these medullary bundles is also an abnormal condition. Then secondary growth and medullary bundle in pericycle region, the extrastellar cambial strip develop. So extrastellar means apart from stele, cambium is developed in pericycle, which produce secondary vascular bundle. Cambium produce conjunctive tissues. What is conjunctive tissue? Conjunctive tissue means the tissues which are present between xylem and phloem. Secondary vascular bundle and conjunctive tissues are present without any sharp limits. So cambium produce secondary vascular bundle as well as the conjunctive tissue. Conjunctive tissues are the tissues which are found between uh, two vascular bundles. But in case of this Acheranthus stem, the cambium layer produce conjunctive tissue as well as the secondary vascular bundles. So phloem of the secondary vascular bundles appear in the form of patches. This phloem is called included phloem and medullary bundles are leaf traces. So this is the structure of your anomalous secondary growth found in Acheranthus stem. Then anomalous secondary growth in Nyctanthus stem. So the outline of the transverse section appears quadrangular. The Nyctanthus stem show a very peculiar morphology because their stems appear as quadrangular, and the tissues which are present outside to inside are epidermis, then cortex, then cortical bundles. Presence of cortical bundles is seen in case of Nyctanthus. Four vascular bundles are present in cortex situated one each in each protruded bulges. Each conical bundle faces its pointed xylem end towards the outer side, that is epidermis, and is conjoined, collateral, open and exarch. Exarch means the protoxylem towards outside and the metaxylem towards inside. Generally, this exarch type of vascular bundles are observed in case of roots, but here in Nyctanthus, Nyctanthus, the same shows exact type of vascular bundle. These bundles may show secondary growth at maturity. So in primary growth, these cortical bundles are also present during the primary growth. Then endoderm is not well developed. Pericycle is in the form of sclerenchymatous patches. Vascular system consists of primary phloem, secondary phloem, cambium, secondary xylem and primary xylem. Primary phloem is crossed and irregularly present in patches below the pericycle. Secondary phloem is present in the form of continuous ring and consists of sieve tube, companion cell, phloem, parenchyma. Then pith is thin-walled and parenchymatous. So what are the abnormalities of Nyctanthus stem? The presence of cortical bundle which are inversely oriented, four in number and never directly connected with the main axial ring of the vascular cylinder. So it is placed abnormally. These are leaf traces bundles. Cortical bundles have been reported in some families such as Cajuarinaceae and Umbelliferae, Papilionaceae and Marlastomaceae rutaceae. So the presence of the vascular bundles in cortex have also been reported in these families. So this is the transverse section of Nyctanthus stem. These are four cortical bundles which are found in four diagonal places, and they are also placed inversely. Then in mirabilis, the anomalous secondary growth occur due to the formation of accessory cambial ring exterior to the primary vascular bundle. So the main vascular bundle have cambium. Apart from that normal cambium, the mirabilis stem have many accessory cambial rings, extra cambial rings in case of mirabilis stem. Then Dracaena stem, anomalous secondary growth seen in Dracaena. So Dracaena is a monocot. Generally monocot lack the secondary growth. So presence of secondary growth in monocot is itself a anomalous thing or abnormal thing. So Dracaena is a typical example of anomalous secondary thickening in monocot. Typically, secondary thickening is absent in monocot. Therefore, secondary thickening itself is an anomaly in Dracaena is a monocot, as Dracaena is a monocot. Dracaena young stem of Dracaena has typical monocot structure like epidermis is single layered, hypodermis is sclerenchymatous. In monocot, the hypodermis is sclerenchymatous, but in case of dicot, it is collenchymatous. Numerous closed collateral vascular bundles are scattered in the parenchymatous ground tissue. Closed means absence of cambium. Then xylem is endark. As it is a stem, the xylem is always endark. Anomalous secondary thickening in Dracaena is due to the extrastellar cambial ring in monocot stem at the cortex. So, in the cortex region, cambial ring is formed, and it also shows abnormal activity. Then during secondary thickening, the formation of secondary meristem and secondary cambium occurs in the inner region of parenchymatous cortex. The activity of cambium is abnormal. How? Because it produces secondary vascular bundle on its inner side only, and parenchymatous cells on the outer side. Generally, cambium produces uh, secondary xylem towards inner side and secondary phloem towards outside. But here it produces secondary vascular bundles towards inside, but parenchymatous cells towards the outside. The secondary vascular bundles are amphivasal, where phloem surrounded by the xylem. So, secondary vascular bundle, in case of secondary vascular bundle, they are amphivasal. Amphivasal means the phloem present in the center surrounded by the xylem. Later, cambium produces more parenchymatous cell to inside that pushes newly formed vascular tissue to the center. The activity and position of vascular bundle keep on changing because earlier the cambium produces parenchyma towards outer side and vascular tissue towards inside, but later on the cambium produces more parenchymatous cell towards inside, which pushes the newly formed vascular bundles towards the center. The activity and position of vascular bundle keep on changing, and vascular bundles are arranged in concentric rings. Secondary ring of vascular bundle is alternating in position with the first ring. Vascular bundle in last inner ring are embedded in a mass of lignified conjunctive tissue. Cork cambium activity is normal and produces cork and secondary cortex at the outer region. So, this is the Dracaena stem, transverse section of Dracaena stem. So, these are the collateral vascular bundles. These are secondary parenchyma. This is the development of cambium ring, and it produces secondary parenchyma towards the outer side, and towards the inside many amphivasal vascular bundles are formed. So this is the transverse section of Dracaena stem. So it is all about your secondary growth or anomalous secondary growth, which are found in different species like Borevia, Bignonia, Mirabilis, Nyctanthus and Dracaena. And these all are present in your SSB lectureship botany syllabus. So I hope you really enjoyed this session and get benefits from it. For more videos, please like my channel, please share with other friends, and please subscribe to my channel Sunanda's tutorial, and stay tuned. Thank you.